So thanks, everyone. I think I bring a little bit of a unique perspective because of the kids. Several years ago, because of my kids, I decided to become a patient advocate kind of lifelong. So even though my daily job is with biosimilars, I do a lot of work in rare disease and I've been doing more and more in it. So anyway, I have a little bit of a kind of a 30,000-foot view on the muscular dystrophy landscape, which is a little bit different. One thing I'm excited about is I used to work in investments. So for about 12 years, I worked in the due diligence group of what was then called Quintile's Legacy. Now we're called IQVIA, and it's kind of a funny term. So the I comes from the IMS, the Q comes from Quintile, so it's IQ, and then VIA supposedly is Latin for moving together. So it's actually, that's how our name came about. So if you see it, it's kind of a weird name, but we're two strong legacy companies coming together. One thing I think about gene therapy is interesting is I looked at it from an investment standpoint years ago. And seeing how far it's come, we're going to talk a little bit about some of the very basics. I'm going to provide you with some of the things just to kind of provide a platform. And then the real experts are here in the panel. So I'm going to introduce them then, and they're going to introduce themselves about some more about what they do. But like Dr. Kelly said, I think this is a pretty exciting area. So if we could go to the first slide. I guess they make you put that up, but I think what's interesting is you know, we do have some of the top leaders in the group here today, so that'll be fantastic for all of us. So a special thanks to everybody. Exonics, Pfizer, Sarepta, Solid, and I've even noticed some other folks have been here from other organizations as well, so thank you for them. So, you know, I tried to keep this a kind of more of a 30,000-foot view, but I have appreciated from coming to these meetings like today is that you guys are a pretty sophisticated group of folks. I mean, I know what it's like having to get two F5 wheelchairs just in the last 6 months, one for my daughter, one for my son. So I do share many of the same things that you guys are dealing with. And a lot of times when I see gene therapy, it gets kind of confusing. So what I hope I can do is bring it down to kind of a level where we can kind of get it. And then when we need to go into something more granular or more detailed, thank you, then we can talk to our experts. So let's just take a couple minutes with this slide and determine kind of what is gene therapy and kind of get it so we can kind of level set everything. So it's an experimental technique, uses genes to treat or prevent disease. Okay, no problem. Dr. Kelly's going over some of the things about actually inserting genes and things when he was talking about some of the different programs that are in different phases of development that you guys all saw and I was privileged enough to join this morning on. So there's kind of three main ways. So, and I was asked to talk about the gene editing. So that's the first one. That's replacing the mutated gene, so the bad gene that causes the disease with a healthy copy, so the good one, right? So that's the gene editing. But then there's another way to do it. You can actually inactivate or knock out that bad gene that's functioning improperly. So that's the transgene therapy. So a lot of this around the gene therapy to me is almost like what I call pharma speak, right? You have to kind of understand some of the logo and kind of understand some of these things so you can kind of get it there. And then the last one is you can actually introduce a new gene to help fight the disease. So that's your surrogate therapy. So those were kind of the three groups. So that's kind of, we'll be talking about one or another of those. When you look at and are evaluating those, you can look and see which one is there. So although, according to the definition, this is just written right off there, although gene therapy is promising treatment for a number of diseases, including inherited disorders, some types of cancer, and certain viral infections, and I put in DMD, that's why I put it in yellow, just because, you know, that's not part of the definition, but it is true for that. The technique remains risky and is still under study to make sure they'll be safe and effective. Gene therapy is currently only being tested for diseases that have no other cure, so it's mostly in cancer or in the muscular dystrophies. So, like I said before, when I worked in investments, I looked at a lot of gene therapies, and I would agree that there was risk. But I have seen some tremendous progress made over the last 15 to 20 years, and it's incredible now that we have some approved, and we're going to talk a little bit about that too. So on the right part of this slide here about some of the gene therapy background, a couple folks said, well, let's take it in kind of the NORD perspective. Let's look at that. And there's actually a nice video that someone had shared with me actually from the CureDuchenne group, which I looked through the whole thing. It's not that long. If you're interested in this and want to learn more about it, it's a nice background because it talks about a lot of things. So I just took out a few bullets that I thought might be interesting for this group. And that is, gene therapy has been around a long time. I remember looking at it in the 90s from an investment perspective. It's becoming more exciting, as I said, because they're now becoming approved. So the FDA is looking at the risk-benefit of these products, and they're starting to approve them. There's more preclinical and clinical data are being generated. I mean, when I looked on clinicaltrials.gov just last week when I was writing a paper, I think I came up with 238 if you use the fully spelled out search term, Duchenne muscular dystrophy, 238 trials. I can tell you that 15 years ago, I can't remember what the number was, but it was a lot smaller than that. Some types of cells are really amenable to gene therapy. But there's a kind of some different things. So the somatic cells, though, that have the 2 sets of chromosomes are, so that's yes, amenable, yes, are to therapy, but not yet the germ cells, so the egg and the sperm, so not quite there yet. Like mentioned, FDA needs to approve all the products, ensure they're safe. It's kind of interesting, I think, that Parent Project Muscular Dystrophy, like CureDuchenne, honestly, I believe are the leaders of all the muscular dystrophies by writing the first FDA regulatory guidance. I thought it was absolutely incredible. And also, the groups that are here and some of your other sister and cousin groups were ones that were actually instrumental in writing the MDA CARE Act. So just incredible. So side effects are expected with some of these types of therapies. We already talked about some immune reactions and how you can do that. I think Dr. Kelly mentioned that to a couple things before. When you're injecting a virus, you're injecting something foreign into a body, it could potentially see it as foreign, and that could cause or elicit an immune reaction. So that's why there's some nuances that we were talking about this morning about steroids and corticosteroids and things like that, because that'll blunt the immune response. So you want to blunt the immune response so you don't get like an anaphylactic or some kind of a bad shock, right? But you also want to maintain some immune therapy so you can fight diseases like a cold and things that we talked about this morning. And one of the things that the NORD mentions is that it's unknown exactly how gene therapy is going to be covered by insurance. So I thought, well, let's give you just like a little snapshot of the first one that was approved in an inherited disease. So there's only been about 3 approved that I understand. Is that correct? There's just 3. And 1 in an inherited disease. So we're just going to talk about that one today because I think that's the one that would be the most applicable for Duchenne muscular dystrophy. So that one just came out in December 2017, so less than a year ago. First gene therapy for an inherited disease, Luxturna, works by carrying a gene into the eyes of patients born with retinal dystrophy. So the retina is the back of the eye that takes all the information so you can see. Doesn't work. Some patients actually reported some dramatic results that they could see images and that they could actually see and things that they had never been able to have before. I mean, pretty dramatic results. If you read about it, I think you'll be pretty impressed because I was. And I just read like 2 articles on it. The cost is not cheap though. The cost is expected to be around $475,000, almost $500,000 per patient. So that's a lot. So when I see gene editing or when I look at a lot of genetic papers and stuff, and hey, I'm a scientist, I'm a veterinarian by training. I mean, I'm a fairly smart guy. But you know what? When I see gene editing and I see stuff like this, I'm like anybody else. I'm like, what does that mean? You know, what do the little scissors mean and what does this stuff mean? And I'll be honest with you, I'm not the guy who knows that. These are the guys that know that. But we have to know a couple of basic things. So like, you know, and that's how we're going to navigate through this and try to understand what the risks are to our kids or our families. So there's 2 primary classes of gene therapy that I'm going to talk about today. There's the ex vivo gene therapy and then the in vivo gene therapy. So those are just fancy terms again, like I said, that's back to the pharma speak. So what that means to translate for you, so ex vivo is kind of outside the body. So vivo means living organism, so ex, outside living, so outside living body. So the cells are modified outside that. It can be done in test tubes, it can be done different ways, modified and then administered to the patient, right? That's one way. The in vivo is directly added to the body and changes the cells in the body. So that's systemic, meaning it can go into like your, into a vein, so it can go and travel all around. Or a local administration like a sub-Q or like a shot in your arm, like that. And then there's the vectors that we talked about it before. So whenever you see those little letters like we did before at the, when we were describing each of those trials, you know, you'll see this AAV and AV, and you're like, what's the difference between that? So the adeno-associated virus, certain type of virus, adenovirus AV, and then there's other things like lentivirus plasmids. The only thing that I want to mention about them today for our purposes is these are vehicles to be able to get the things that we need to change the genes into the body, right? Because they're going to have something, there's going to be an action to have to do something, and these are those vehicles. I'm not going to talk about them too much in there, although I will show you a slide on a couple of different ones so that when you see those names come up and it's associated with a certain type of way that the sponsor is trying to get these genes delivered, you'll understand a little bit better, hopefully. And basically, the vector type, I want to leave you with this, that's why I underline it, is that the type is based on the desired type of gene replacement duration, right? So the concern like 15, 20 years ago, I remember when I was reviewing these projects, is I would see a gene therapy and I would see, I had the privilege of looking at like FDA guidance and stuff like that, which is generally proprietary. But they would have things like, you know, a 15-year monkey study because they wanted to make sure they had the comfort in it before it was introduced into humans, right? So they want to make sure they look at it for a long time. Well, if you're a company and you have to do that, that's quite a bit of commitment you're asking for a company to do. And then the gene editing can be used in both the ex vivo and the in vivo. And then just a little viruses 101. Just remember that viruses that we get even in colds and stuff like that, they naturally use our DNA and RNA, right, to replicate. So there's different ones. There's RNA ones, DNA ones. So they do that naturally. So that's just something that happens to every one of us is going to get a cold probably in the next 12 months, right, some kind of cold. So that's a normal thing. So we're just taking them now and trying to take some of those natural attributes and putting in it like an artificial setting in order to get the genes manipulated that we want to, usually typically around that dystrophin. And then some viruses can cause cancer. So that's why when you see certain types, they can actually do that. So papillomavirus is one that can actually cause like a lump on you. Maybe you've seen skin or known of people that have had that. Or there's papillomavirus will also cause cancer in women. So I'm not going to go over each one of these, but just to give you like a little thing there. So like, so there's some kind of things that are like not viral that could actually get the gene in. So those are like plasmids, which I know those mostly from like bacteria. So they're able to manipulate and use chromosomes. That's what they do. Or you can stick it to some fat. So liposome is just a fancy name for fat. So that's that. And then the adenovirus and the adeno-associated virus, those are biggies because those are used a lot in gene therapy for DMD therapies. And those are just two schematic pictures of what it might look like in a 3D setting. And then there's viral DNA, and then we were talking about before, just naturally occurring viruses. There's viral that are RNA. So those are two different types, the lenti and the retro. So just to kind of give you a little overview on that. A couple of more things on the ones that you'll see more in the DMD space, and that's the adeno-associated virus. And that's used more for long-term gene replacement. That is approved for gene therapy use. It's often used in vivo, so inside the body. It infects dividing and non-dividing cells, and it's kind of the vector of choice for non-dividing cells. One thing, though, on kind of like the safety side is just remember that it does not integrate into the cellular DNA and it doesn't replicate. So it's not going to do more than what it's supposed to do. It's not going to keep doing things afterwards. And it's the vector choice for the AAV for long-term expression in things like muscle. So for like Duchenne muscular dystrophy, also skeleton, brain, heart, and heart also for DMD. And the length of expression is unknown, but it could be from anywhere from 2 to 10 years. So you can see there is still some uncertainty around these. So let's go over just a couple of things of what would make a good like gene therapy target. And I'm just going to just touch base on a couple of them. There are more, and there's a lot more that are very specific and detailed for the different types of therapy. So if you have a question on those, again, I would ask our experts on that. But from my simple kind of perspective here, a couple of things would be nice is to have a disease physiology that has a clear disease mechanism. So we know about dystrophin, we know about the problems with that. If you can identify a single identifiable genetically driven flaw. So this is more amenable to, say, the DMD space than, say, FSHD, because that's thought to be one of the thinking about FSHD is an overexpression of a protein called DUX4. And that's not all fully well known. So that would be less amenable to this type of therapy, whereas there's a lot more known about Duchenne and the genetics around that. So that's good. That's a really good thing. Also, a high unmet medical need. I don't think anyone would argue with that. So there's, but we have to have a balance of the significant quality of the life impact and because they have limited survivability, that's why it's restricted to just using in cancer and to DMD right now. So there has to be like no or limited effective treatment options. The last one that I want to leave with on this slide is the curative potential, because I think that this is the most exciting thing. I helped start a patient advocacy group in North Carolina for the FSHD Society about 3 or 4 years ago. And my daughter just took over it this year as the president. And they're watching CRISPR and all these other things. They have the same type of group like you guys do. And we're all watching this because I think the last one is the curative potential. I think it's what I've seen in life is anything that has high risk can have high reward, right? So I think there is the potential there. And they're starting to enter the clinical development. You've seen the bunch of them. I think Dr. Kelly's done an outstanding job of giving you an overview. I have seen several talks, and that one was really nice. It provided the different types. It told you what they were modifying, what they were doing, and even some that didn't have anything to do with gene therapy or gene editing, but he kind of went over all the other ones for DMD. And there's 3 that I'm aware of, the TALEN, the ZFN, or the zinc finger nuclease, and then the CRISPR. And the CRISPR, I've actually had an opportunity because I sit on the board of visitors at NC State, and I've seen several people in the preclinical models looking at CRISPR for DMD. So even though that one is kind of what my colleagues who helped me develop these slides, and they actually run what they call the Gene Therapy Center of Excellence at IQVIA, said it's kind of the most hype because everybody hears about that. I mean, I couldn't believe that you could actually go online and get a CRISPR technology, which is basically just like a scissors for cutting DNA, for about $75. So to me, that's like, whoa, that's like pretty inexpensive. But one of the things that he told me just to mention to this group is that the selectivity will be needed to be improved for the clinical use. So we do have some folks who are working on CRISPR technologies. You can learn more about that because my experience is limited with that. And this is where I took that figure out form just so you could see. So the ZFN is the one on the left, how that works with the scissors. The talon is the one how it works there, and then the CRISPR is the other one at the end. And the scissors are how it cuts these pieces of either DNA or RNA. And all those little letters stands for the different DNA. So guanine, cytosine, adenosine, you can see tyrosine, you can see the little letters, and that's what they mean there. But I mean, like I said, when you're starting to talk to that level, or you look at a lot of papers like I do, and I'm trying to translate, I'm like, so what's the bottom line? You know, what does it mean for me? Because that can be hard with some of these genetic things. Our company gets involved with some of the gene therapy development planning. So I'm just going to talk about a couple of things with that because it really requires a special development planning consideration. It's like with the regulatory, there's like kind of like a pre-IND, then there's the pre-IND meeting, and you have these special meetings with the agency. There's the long-term follow-up I was talking about before as far as like monkey or primate studies and things like that done. And then this kind of ability, or however you want to look at it, inability to kind of redo it. Like once you give it, it's kind of done, right? You can't take it back. We've talked about some things that shows you that it's potentially can be curative and durable, so it could last a long time, but it could be costly. You know, the first one coming out almost $500,000. There's some outcomes focus and things that you need to consider. There's quality of life issues we need to consider. PROs is just a fancy word for patient-reported outcomes, so we need to include the patient in all this. We need to also have a reduction in the overall cost of care. One thing I'll tell you interesting is that our company, like I said, worked with the MDA. When we were working on them, we did what we call a landmark study. It just came out this week. If anybody's interested, I'll be glad to see this here. And the one thing that struck me in there that I want to share with you is that about 250,000 people, the report said, has a neuromuscular disease. And the cost associated with that, so meaning all the caregivers, the parents, maybe buying another home because like we had to buy a home from 2-story to 1-story because our kids couldn't go down the steps anymore. If you take all those costs into there, for those $250,000 kids in the US annually is $46 billion. Now, 250,000 people driving $46 billion worth of cost isn't time for innovation and disruption, and I don't know what is. So I really think that gene therapy could be one of the answers to this and could be potentially a big one. I'm not going to talk about the manufacturing for that, but I will say that there is some value there that folks may have questions, and maybe the experts can answer that as well. I'm just not familiar with that. So with that, I would like to invite each of the panelists up. So it'll be Dr. Jesper Gromada. Could you come up, please? Steve. Dr. Allison Craney. Dr. Genevieve Laforet. And Ms. Tara Moorehead. So Dr. Gromada, he's the Chief Scientific Officer of Exonics Therapeutics. And Dr. Craney is the Associate Director of Patient Advocacy for Therapeutics. Dr. Laforet is the Senior Director in Clinical Research and Development for Solid Biosciences. And Ms. Moorehead is the Director of Clinical Research for Pfizer. So I'd like to turn it over to them to introduce themselves and some of the projects that they're working on.
I'm the first one. I'm Jesper Gromada, and I'm totally new to all of this. I've only been with Exonics for 4 weeks. But I'm very happy to be here. I worked previously in a company in New York called Regeneron Pharmaceuticals, which is a big biotech company with a lot of autoimmune diseases. I was very happy, but when Exonics approached me and they told me about the work they were doing, I was totally sold. I had to do it, so I came here and joined and moved back to Boston because I used to work at Novartis in Boston before I moved to New York. So everything that I will be presenting, I have not done any of it, so I cannot take any credit for it, but I'm still very excited about it. And so this is what it is. So at Exonics, we are developing gene editing therapies, and we're using CRISPR-Cas9, the famous thing. And I will tell you a bit about this in a minute. To treat first the Duchenne muscular dystrophy patients. And I'll kind of walk you through very simply what we're really trying to do. Here we have the dystrophin gene in normal, dystrophin protein normal. This is from dogs. This is the respiratory muscle of the diaphragm from the dog, and this is the dog heart. The dystrophin is staying in green, so you can see there's a lot of the protein. Okay? Here we have a mutated dystrophin gene. There's a stop codon in, which means that the gene is not being translated. There's no protein made. You can see there's no real green. There's a tiny bit of green here. That's because it's a background, but there's no protein made. All right? So what we're using is the CRISPR-Cas9, which is literally a scissor. It's very specific when you do a lot of work to optimize it. So what we have done, we have made a guide which tells exactly where we want the cut made. Very specifically, at one side, at the gene. And then we have the Cas9, which makes one cut in the DNA. And when we make that cut, there's a repair that basically causes the mutated gene to be a corrected dystrophin protein. And here you can see this is in a dog. After 8 weeks after the treatment, you can see how much green, basically, dystrophin protein that is here. Very importantly is that the size of the protein is nearly normal. It's 99% of the normal size. It's not a small truncated version. It's 99%. What is also important is that after 8 weeks, we have seen up to 90% restoration of normal levels of dystrophin. So this is our Exonics pipeline for Duchenne muscular dystrophy. We are working on different exons. We are at different stages. We are hoping soon to be in the clinic if everything continues to look as promising as it does now. So this was my 2 minutes plus.
Hi, everyone. My name is Allison, Craney, and I work in patient advocacy at Sarepta Therapeutics. It's really a privilege to have been with all of you today. I actually spent numerous years working in a cancer cell biology lab before joining Sarepta and was working with some of the same viruses that we are using today and some of the very techniques. Was in the department in graduate school when one of the canonical CRISPR papers was published. So it's just a real privilege to be seeing all the progress that this has made. So I'm excited to tell you a little bit about Sarepta today and then try to help answer any questions that you have. So Sarepta has historically been known as an exon skipping company, and we do have a number of exon skipping pipeline programs. But today I really want to focus on the 4 programs that we have in partnership in gene therapy and gene editing. And I do want to highlight this is in partnership. So we've been very, very privileged to have wonderful partners in this work. So we have 3 gene therapy programs. So 2 are microdystrophin gene therapies, one in partnership with Nationwide Children's Hospital and another in partnership with Genethon. The Nationwide Children's Hospital is currently in clinical phase, where Genethon program is currently preclinical. In addition, in a collaboration with Dr. Kevin Flanagan's group at Nationwide Children's Hospital, we also have a collaboration looking at a surrogate gene therapy approach with GalGT2 that's also in clinical phase. And then we are also excited to be partnering with a group at Duke University in gene editing specifically for Duchenne muscular dystrophy. And this is much earlier in the research phase. So as I said, we only had 2 minutes, so I'm going to just quickly describe a little bit about our most advanced clinical program for gene therapy, which is the microdystrophin gene therapy in collaboration with Dr. Jerry Mendel and his group. And I really hope that if you walk away with one thing from my talk today, that you recognize that there are typically up to 3 major differences among programs in gene therapy. And that can be the viral vector, the promoter, and then the gene that you're actually trying to get into patients. And this is kind of a silly analogy, but I love analogies, so I'm going to try it. So I kind of think about these as like a CD player where you're trying to play a CD of greatest hits from your favorite band. So let me try to explain that. So the key aspects to remember from that analogy would be the CD player, the CD of the greatest hits, and the play button. And you can tell me later whether or not this analogy is good or if I should work on another one. So first, the viral vector. So in my analogy, I kind of think about the vector as being like the machine, the CD player. So it really has all of the machinery that you need to kind of produce the music, but in and of itself, it's not producing any music. And so for Sarepta, we are using a virus. It's called AAV, and all the companies today that are speaking are using an AAV-typed virus. And we are specifically using a virus called RH74. So the RH actually stands for a rhesus monkey, and the number is just, usually we number things as scientists in the order that they're found. So rhesus monkey was the origin, and then the number 74. And we're excited about the fact that this was a vector that was not isolated from a human host, and we hope, and we're still exploring right up to this day, and we will continue to explore whether or not having a virus vector that's not derived from humans would potentially lower the immunogenicity factor. So that's something that we're excited about and we're currently exploring. And what we're also very excited about for our RH74 vector is that our preclinical work and some of our very, very early clinical work just demonstrates delivery to all of the muscles that we'd really want it to get to for Duchenne muscular dystrophy. So this is skeletal muscle to the diaphragm and also to cardiac muscle. So that's the viral vector, that's the CD player. And then kind of moving on to the transgene, or the gene that you want to get to patients. So in this case, we're looking at microdystrophin. So imagine your favorite band is the Beatles. The Beatles have produced a lot of music, and you can't fit all of those onto one CD. And that's kind of similar to dystrophin. It's a huge gene, and it's a really important protein, and you actually can't fit that entire gene into your CD or your CD player, which is the viral vector in this particular case. So what you have to do, and what all of our companies have had to do, is really look very carefully at the dystrophin gene and try to very, very precisely select which pieces you were going to keep or which of your favorite songs from the Beatles you were going to put onto that CD. And so our microdystrophin, you might hear microdystrophin from different companies, but even though they have the same name, they're actually different genes because each company has used a different process to try to determine which parts of the gene to move into the viral vector. So our microdystrophin we're really excited about, and it's been developed through very precise genetic engineering based upon clinical observation of a patient who had Becker muscular dystrophy. And then so you have a CD, and you have a CD inside a CD player, but you need a play button. So the play button in this case is a promoter, and the promoter, we are using a muscle-specific promoter. It's called MHCK7, but the name really doesn't matter. Basically what this means is that no matter where the virus goes, you're only going to play the microdystrophin when you're inside a muscle cell. So we think that's really important. And we're really excited about that specificity that having a muscle-specific promoter allows, and also the fact that this muscle promoter in our preclinical studies has enabled widespread microdystrophin expression across all the muscle types that I mentioned earlier. So you can tell me later what you think of my analogy, and my feelings won't get hurt. I can take it. So I didn't have time to talk about our other programs today, but we do have a website, DuchenneGeneTherapy.com, and we're currently adding a lot of content to this. But if you want to hear more about our program today, or hear actually just from the experts themselves, the doctors who have done all of this early work and their teams, I'd really encourage you to visit this website to learn more about those programs, and then the other 3 programs that I didn't have a chance to talk about. And then just, of course, thank you so much to all of you and to CureDuchenne for hosting this wonderful day.
Good evening, everyone. My name is Jenny Laforet. I'm Senior Director of Clinical R&D at Solid Bio. It's my pleasure to speak with you today, and I want to thank the organizers very much for the opportunity to be with you tonight. I have to thank Allison for creating a wonderful analogy about some of the things I'm going to be talking about, so she's already done some of the heavy lifting. But let me proceed to just tell you a little bit about our gene therapy program, SGT-001 for microdystrophin gene transfer. The lawyers make me put up this text up here. I supposedly have 2 minutes, so I'm not going to dwell on this at all, but move on with all due haste. I also want to introduce you a little bit to Solid Biosciences. We're a young company founded approximately 5 years ago, and really have this unique singular focus on Duchenne. As many of you already probably know, it was founded by parents of a young boy with Duchenne, one of whom is in the room tonight, Annie Gunott. So the sense of purpose is very deep. And the effort has been made to really create a Duchenne Center of Excellence, to gather the best scientific minds to advise us, to help us design our programs, to develop a leadership team that is experienced and really can bring our mission to fruition. And also a 360-degree approach to the disease. This is not about a particular technology or a particular therapeutic approach, but really scanning for the most promising approaches that are really going to make an impact in the lives of Duchenne patients. So the focus of tonight's discussion is our lead clinical program, that is SGT-001, which is an AAV microdystrophin gene therapy candidate that really is attempting to address the underlying cause of Duchenne, that is this dysfunctional dystrophin gene, and how can we effectively restore that function. I just have to emphasize, this is so important to everything that we do, that patients are really at the heart of it. This is what it's all about for us. So what is gene transfer for Duchenne? And Dr. Huml really laid the groundwork for this, and it was supported by speakers after him. But really, it's the three essential elements that are important that we have to be thoughtful about. What are the best components to assemble in this potential therapeutic approach to really make it work? So one is the gene. We've already talked about that, how important it is to have the right elements of the native dystrophin sequence that will have the right kind of function so it'll do what it should be doing. And the other one is the promoter. Kind of think of it as what drives how, where, when, how much of the microdystrophin protein is being made. And then the vector, that's the vehicle, that's what delivers it to where it needs to go. So putting all of those elements together is what makes your gene transfer agent, candidate drug, whatever you want to call it, and that's what the patient will get. Carries the whole package into the body to do what it should be doing. So in the case of dystrophin and Duchenne, this has been talked about before. This is a huge gene. There's no way to pack. It's really the largest gene in the human body. To be able to fit that into a little AAV vector just can't be done. So it's a matter of pulling out those key elements that are really important for the function. It's like the Reader's Digest abridged version of the dystrophin. Pack that into a small piece of DNA, a small gene that's going to fit within the limited carrying capacity of an adenovirus particle, and then make an abridged version of the protein, but one that has those critical elements to still really do what it needs to do. And we've used a adeno-associated virus number 9, again numbered in the order of discovery. But AAV9 is widely used in gene therapy and gene transfer. It's used by other microdystrophin programs as well as other genetic programs that are in the clinic now. It's one that's very well characterized. There's a lot of experience with it, scientific experience and now clinical experience that's growing. And also, but the AAV9 also has the right kind of packaging capacity, and we understand the biology of the outside of the gene, the outside of the capsid well. So the composition of SGT-001. The graphic on the top shows this huge, large, natural dystrophin protein. It has lots of different domains or portions of the protein that are important for function, but some of them are repeated. So are they redundant? Are they all needed? The idea is, again, to boil it down, to condense it down to a working version, but that has just the essential things that it needs. So SGT-001 was designed to retain key components of dystrophin. And importantly, one portion, the neuronal nitric oxide synthase binding domain. One of dystrophin's roles is to bind other proteins, to keep them in the cell where they need to be, so they can do their function. And one of those important proteins that it binds and helps work is this nNOS. So this is thought to be an important kind of regulatory function of dystrophin that we wanted to retain in microdystrophin. So on the bottom, you can see a graphic of this microdystrophin construct, the one that we selected for further study. And this was after scanning and testing other microdystrophin potential candidates and picking the one that really had the best activity. Also, we talked about the promoter, how this drives the whole system and makes the microdystrophin express the instructions it's supposed to express. And this is the CK8 promoter. This is, again, a muscle-specific promoter that restricts expression of this microdystrophin gene just where it needs to be, skeletal muscle and cardiac muscle. That's where the action is. And all of this is packaged into the AAV9 vector, as I described earlier. So those three components, the gene itself, the promoter that's going to make it expressed where it should be expressed, and then the vector that's going to take it where it needs to go. So those are the components of our SGT-001, and it's probably way over 2 minutes, so I'll stop there and pass it along, but we'll have more chance to ask questions afterwards.
Hi, my name is Tara Moorehead. I'm a director of clinical research at Pfizer and the clinical lead of our Duchenne gene therapy program. I've been involved in working in Duchenne muscular dystrophy within the clinical space in clinical trials for going on 5 years now, and it's an honor and privilege to be here. We also have this lawyer thing, safe harbor, and because we are a publicly traded company, so as far as any forward-looking statements. So fortunately, I got lucky because I'm the last presenter, so I just kind of get to summarize and build upon the other presentations. But our gene therapy product, or investigational product, as we like to call it, is labeled PF069-39926. We love making up really catchy names. It used to be BMB D001, and that was when it was developed by a smaller company, Bamboo Therapeutics, and the brilliant minds of scientists such as Zhao Zhao, Jude Samulski, and others, and for which we received a significant amount of seed funding from CureDuchenne, and Pfizer was very interested in this construct, which I'll talk about in a second, but we purchased Bamboo Therapeutics, and they're now a wholly owned subsidiary in 2016, and we've been able to bring it into the clinic. But just a couple points as far as our particular gene therapy. We have, we call it a mini dystrophin. I think I've said before, it's kind of tomato-tomato in terms of micro versus mini, but it is a different sequence. And this was based on the one that we use, which is at the bottom. It's DYS, the 3978, and that was developed using a patient with a mild form of Becker muscular dystrophy, so his sequence as a model, and to try to pick all the domains that seemingly reflected or were very similar, and that's in the middle column, that could fit into this AAV9 packaging. As we said, we're limited in terms of the capacity, so we tried to pick the ones that seem most critical and that could at least potentially, if it works, instead of having a traditional maybe severe or mild to moderate severe Duchenne, we can maybe change the phenotype or the clinical manifestation to more of a mild Becker patient disease. And so I think, given the time constraints, we also are using the AAV9, just like the solid Biosciences, as our capsid, and we picked that because of the, it seems that it's not as leaky in terms of it gets to where it needs to go. It's non-pathogenic, which is great, but also in terms of the global transduction in both the skeletal and cardiac muscles. And we also have a muscle-specific promoter. It's a human muscle-specific promoter, so we have all the components, as Allison talked about, in terms of a CD player. And here you can just see. This is a build slide. I think Dr. Kelly went through really quickly and probably did a much better job than I will have time for. In terms of our current ongoing clinical trial, this is a safety study. We're focused primarily on safety, which is really important, so it's a smaller study. It's our first time being in humans and in patients, but just to tell you that we are looking at boys who are 5 to 12 years old. They do need to, I think we'll end up talking probably during the panel in terms of neutralizing antibodies, so we have a test for that, and they have to be negative for neutralizing antibodies to the AAV9 capsid, and so that's part of the eligibility criteria. But the bottom line is, for us, we aren't excluding any patients based on mutation, which is great. And we are staggering our dosing. I think all of us needs to in gene therapy in terms of pausing after each patient receives a dose. Every patient just gets one dose. and just making sure that there's no safety findings before proceeding. We're looking at 2 dose levels, and we're looking to enroll maybe about 12 patients, but that just depends on the data that we're seeing and how much, and the size of the boys that we're enrolling. And I think I'll keep it short and sweet there, but just thanks to the community. Right now, we have 3 patients who've been dosed in our trial, and they're doing well, and we're looking forward to completing the study. There's been incredible interest, so we feel very humbled that the families are willing to participate in this very early. We're very much in the early days, and I think that's important and worth mentioning, but that there are families who are willing to participate in these trials because it's the only way we can progress forward. And yeah, we're excited, so thank you very much for the opportunity.
I think next we're going to open it up to questions here, so from the audience. So if anyone would like to ask a question, go ahead and raise your hand. I know some of these can be a little bit daunting, but here we go.
When you say skeletal muscle and cardiac, so do you die?
Yes.
Yes.
Yes, okay, sorry.
Yes. So the question was, does that mean cardiac muscle as well as diaphragm? And the answer is yes. It does affect both. Next question.
I have a question for the panel. You stated there before with the CRISPR gene editing, you should have stop codon read through. We're looking that this is going to be able to be utilized for any type of stop codon.
We don't know if it will apply to all exons, but what we can say is we have already proof of principle for 12 exons. So our single cut technology will at least work for 12 exons, that we know. Will it work for more? We don't know. We haven't tested that. But 12 exons will span a large proportion of all patients. So we're pretty optimistic about this.
Thank you so much, panel. I've got 2 questions. One's regarding the Exonics CRISPR technology, but instead of Cas9, what about that CPF?
Yeah, so we are working with all kind of, she's asking basically, there are different types of scissors, different ways of cutting the DNA. We have had tremendous good luck and experience with the Cas9, so we feel that we're getting basically in heart, for example, in the respiratory muscle that somebody was asking about, which is a big problem, we're getting more than 90% correction. So I don't think we can do much better. But for other neuromuscular diseases, we will maybe be using other scissors with different properties, basically.
Because that brings me to the second question, wasn't that talk about immunogenicity to Cas9? And so is that the same problem we're going to run into with the immunogenicity for gene therapy?
Yeah, so that's a very good question, and this is something that is coming up now, and people are debating about basically what she's asking is that there are some studies that are now appearing that there could be immunogenicity to the scissor. And this is something obviously we take serious, the whole community takes serious. Our stand is that there are reports out there that are saying it's really not a problem, and then there are other reports saying it's a big problem. We don't know ourselves how big of a problem it is, and we will of course monitor for this. I think there will be probably immunogenicity in different degrees for all the different types of scissors. We have not seen much immunogenicity overall with our approach in the dogs, so we're pretty comfortable with this right now, but it is absolutely something to look out for and monitor very carefully. No question about this. Having talked about immunogenicity, you can also then think if there's immunogenicity. There's several types of immunogenicities that needs to be considered for the AAV. And we're using the same as most companies, not all the companies. But this is definitely a problem across the board. And that's the reason why you can only give one administration. The other type of immunogenicity that we all need to consider is to the dystrophin protein itself. Because the body is suddenly making a protein that has not been there before. And that could be viewed as a foreign protein to the body. So that's something else we need to monitor for. And then the third part was what was brought up about, in our case, the scissor, basically. So, but these are all things we need to monitor for and just keep an eye out for and keep going. But there are approaches, sorry, that was the last thing, the scissor, and then I'll keep my mouth shut. There are ways of basically eliminating the scissor. So there are a lot of people that are now working on approaches where we're expressing the scissor, we're repairing, and then we're eliminating the scissor, and in that way, it may not be a problem. So we're working on this too.
I was wondering, I know that Michael had said that the CRISPR technology might not be, available for 5 or more years. If we put one of our children into a clinical trial with the microdystrophin, are they going to be able to do the CRISPR once it's available? So it's either or.
Do you want
yeah, please go ahead.
It's either or, because, and the reason why it's either or is because we are all using the same delivery, the AAV, and that's the problem. So if you're enrolling a child for one of these approaches, then you cannot use our CRISPR-Cas9 approach. It's just not possible, not because of the mechanism, but because how we deliver the protein or the scissor.
Good evening, everyone. My question is that a lot of these clinical trials are based on the, in their inclusion criteria that the kid should be on steroid, right? Mm-hmm. So I'm planning to have my kid on a steroids actually. My question is that I came to know that most of the clinical trials do not include a DMD kid if he has any behavioral, cognitive, or social impairment. He may not be a part of the clinical trial because they will exclude him. So I was thinking that does gene therapy trial also considers the kid having a behavioral and cognitive issues? Because my son, for example, he may not be able to follow the commands that you are going to tell him, like, stand or walk or climb the stairs. He may not be able to do it at this age. He's a 4-year-old with the deletion of 63 to 67, which is, I don't find any person with the same deletion. I found there is a duplication, but not the deletion. So I'm thinking that that was one of the hope that if I start with the steroid, down the 5 years, I might get a gene therapy trial opportunity, but it seems that he may not be eligible because of this, the criteria that a lot of these companies are putting forward. So I heard there is something called compassionate use. Does all of your company considers this, or is there something else that I can look forward and approach these companies to consider the kid like mine to, will be eligible at somehow for a clinical trial?
So I can say, I appreciate what you're saying. I think when we're at this very early phase of development, what we're just trying to do in terms of an experiment is eliminate the variables, right? And try to find if we can measure a difference and have it not be maybe skewed by something else, like maybe inattentiveness, which I struggle from, things like that. And I think the idea is that ultimately, if we're able to demonstrate, because right now it really seems that we can agree that beyond just seeing the expression of the mini or microdystrophin gene in the muscle cells, we're also looking to demonstrate a clinical benefit. Mm-hmm. And until we can do that, I think we're maybe a little bit more strict in terms of our criteria. But mm-hmm. the idea being that if we're successful, then it wouldn't necessarily be limiting in terms of other behavioral or mm-hmm. challenges, cognitive challenges, because we've already demonstrated that there is a clinical benefit. Does that help? And I think down the line, maybe we would have compassionate use, but I think we have to first demonstrate safety at least, and kind of get that, and then show some signs of efficacy before we.
Okay, so basically we need to wait for the FDA approvals, and then companies will think about compassionate use, or it will happen before the FDA approval for gene therapy?
Wait, I'm sorry, I didn't hear that.
So I'm thinking that how
You have
soon
to
I
get
can
approval
expect
for-
the compassionate use of this trial.
Or wait, maybe till it goes to market. Just depending, I guess, because I think each of us might be, our approach, our strategy in terms of development to try to get to register the drug with regulators might be a little bit different, so that might also vary. But I think at this stage, that might, just because it's so early, we're really being more stringent in terms of the eligibility criteria. So,
thank you so much.
Sorry.
But I guess that's, if it really works,
Yeah,
then the time until there could be
of course.
treatment made available will obviously be shorter. So it all hinges on how good the clinical trials are, right?
My question is, as a parent, is that something that we can do, for compassionate use maybe? Or it is something nothing in the hand of parents?
The one thing for us, and I can only speak specifically for us, first, thank you so much for sharing your story, because it's really powerful, and I think it's really important for us to hear. I just want to say two things, so to try to answer as best I can both your questions. The first is that we recognize, at Sarepta, we talk a lot about the fact that there are not, the clinical trial, they're small clinical trials, and they're moving rapidly and slowly at the same time, if that makes sense. They're moving slowly in that the trial itself is moving rapidly, but it's small numbers of patients. And we know that, as I'm sure everyone on this panel does, that we need to move as fast as we can to get to the point where there's access for everyone, so approval and access. So I just want to say that there are a lot of young men and affected individuals who aren't part of the trials, and that's something that I think I can speak for all of us to say that that's something that weighs on us heavily and that we're thinking about a lot. I would also say that for compassionate use, one thing that's a little different about gene therapy is that the manufacturing of gene therapy, we call it manufacturing, but when I worked in a lab and was making gene therapy, it never really occurred to me to think of it as manufacturing, because that always made me think of kind of like a factory belt where you see things kind of getting churned out, if that makes sense. But the process of making the virus is difficult, and it's limited in how much you can make at a time, basically, without getting into too much detail. And so for us, that's been one reason why compassionate use hasn't been something that we could entertain for gene therapy, because we quite literally need the amounts of the virus that we're making to use the clinical trials. But I did just want to start by saying thank you and end by saying thank you, and that I'm sure it's something that we're all thinking about.
Something we think about very, very carefully as well. I think it's important to approach the idea of broadening access from a very responsible perspective. As Tara alluded to, this is such early days. Establishing the safety is absolutely paramount. And then also understanding what the potential benefit might be. Until we have a really good sense of that, it really wouldn't be right to make it more broadly available until we really understand what the drug is about and what it might do in a prospective patient. So I know that we share a tremendous sense of urgency, but also a sense of being careful about how we approach this, and that we go forward with broader access in a responsible way.
Thank you so much. Yep.
As I told you, I've just been here 4 weeks, but I've been in the pharmaceutical industry for a very long time. My day number one, I looked at the clinical plan. I said, this is not aggressive enough. We're not doing enough. We are too cautious. And this has been, every day, this has been what I've been saying. We had the scientific advisory board, which were some of the same people on that are doing the clinical trials. They're on our tour. This is number one thing on my agenda, to talk to them, challenge them, and see how we can speed up things, how we can be more creative in the clinical trial. I want to think out of the box and see if there's ways we can do this. At the same time, as that was pointed out, we need to establish, to prove to the regulatory agency that our therapies are working. So it's a compromise between being aggressive, but also do it in a way that we can demonstrate that it's working. And that's the fine balance.
Thank you. I really appreciate that.
I think you raise an excellent point. I think what's, in addition, besides the patients and making sure that safety is well established, because I think it only takes one incident for the risk of everything getting shut down or coming to a screeching halt. And so that's the other side that we're constantly wrestling with as we're entering into this, because we are kind of on the frontier in many ways. I think what's also brilliant is that we're all competing with each other, and I think competition's awesome. And so if that's not also an additional motivation, beyond obviously the patients and these boys who are dying, that we're all competing against one another, I think is a good motivator, because we all have the same goal. And so I think that's another reason, yeah. But we do end up spending many sleepless nights worrying about whether we're doing things the right way and we're designing things the right way and we're thinking the right way about our approach. I think that's fair to say.
I'd like to just add something too from my own perspective, and that is that from looking at it from kind of the outside, about 8 or 9 years ago, as I had looked to see when the Europeans had developed their DMD, kind of like the analog of the FDA, their regulatory guidance back in 2011, and we didn't get it until 2014. And the urgency is so great that patient advocacy groups basically had to take it in their own hands. If it wasn't for PPMD and CureDuchenne and others like them, we wouldn't have even had FDA regulatory guidance. And what regulatory guidance does is it does a couple things, but one is it de-risks programs, but there's still a lot of uncertainty around the gene therapy. And I don't know if you guys knew this, but just an encouraging thing, and I actually forgot to mention it in the talk, but there's been 6 different guidances that came out on gene therapy this year. So it's trying to untangle some of this confusion, and it is taking more time. But I'll be honest with you, as a parent with 2 kids and seeing the progressive nature of in my own family on a daily basis, I share your sense of urgency. I think it is hard too. I was at a meeting with the FDA going over rare diseases in general, and it basically said that in this type of situation, people are willing to accept a lot more risk than they would in another thing. So one thing I have seen the agency do, in my own mind, is companies from the early side trying to negotiate to get it out on the market. And Dr. Kelly alluded that earlier, saying maybe if this product works, you could get out like on a phase 1B, which you would never see that in certain other indications. But given the risk and the life-threatening nature of this disease, what else could they do? So I think we are starting to see some things, even with Janet Woodcock, with recent approvals and things. There are things on our side. It's not as quick as we want, but I do unfortunately kind of agree really that we do have to do it within the regulatory boundaries and the other boundaries of safety that we live in, unfortunately.
That's one more thing that we all need to kind of also acknowledge, and it was kind of alluded to, is manufacturing is a problem, and everybody's doing what they can, but there's limited capacity, the technologies are outdated, and everybody's trying to fix this problem, but it is a problem. So even if we had accelerated approvals and everything, it wasn't sure that we could actually produce enough material for delivering, and that's just a fact. Everybody's doing everything they possibly can, but I want to also manage expectation, because we could push and push and push the regulatory agencies and do clever clinical trials, but we may actually not be able to sustain everybody for treatment. And it goes for all of us. So it is a fact, and everybody's trying to do it, but this is another limiting factor.
Yeah, I just had 2 questions. One of them is we see 99% of the length of dystrophin protein in dogs. I'm wondering if there's a analog to that we can look at in other drugs and see if it works, the same way, the same effectiveness.
So yeah, that's a really, really good question. So what I can tell you is that we have made human cells, muscle cells from patients, and there we see the same 99%. So you take it for what it's worth, but it is really cells that are taken from an DMD patient, and we have made them into heart cells. And there we see exactly the same very efficient correction and the same kind of 99%. So that, again, is another, I didn't talk about it because I had 2 minutes, but that's another kind of part of our data package that gives us hope that this will really also be the case in humans.
And then if I can ask a quick follow-up question about, I know you took the dog from, when it was born in early stages, and for people that are in the later stages and there's already the degradation of muscle cells, is there any repair mechanism?
Yeah, that, again, a really, really good question, and I appreciate you bringing it up. So this is something I want to do, but the dog that is 1 month old when we gave it, That's actually a fairly advanced disease stage. It's not a 15-year-old boy, it's not, but the dog does have a fair amount of disease progression. But we want to try to treat older dogs and see how much recovery we get and how much functional improvement. So this is definitely on our things to do, absolutely. And I would love to be able to see dystrophin protein being made to some extent, I cannot say how much, in older dogs and hopefully in boys too.
Thank you for all of your time.
So my question is about the redosing of genes. So I understand that we might need to redose the kids after 10 years or so after the gene therapy. So the question is, if we do the gene therapy from Sarepta, which is using AAV74 virus, and then after 5 or 10 years, if we need redosing, can we use Pfizer or Solid, which are using AAV9? And I want to know your AAV version also, the CRISPR-Cas9.
Yeah.
I didn't hear the second part, but for the first part, I think that's an outstanding question, and I think we don't know exactly how long the duration of gene therapy will last, and the true answer is probably that it's very different based on who the individual is and what age they were dosed. And I think there's a lot to learn on that, but I do think that there are a lot of people who are working on the redosing question. So I think we don't have an answer today to redosing, and we don't have a definitive number of years to say for duration how long it'll last. We know that gene therapy is likely to need a second dose or is likely to need a second intervention at some point. And I think and hope that all of us are, as well as many of you and some of the ventures that CureDuchenne is funding, thinking about what is that redosing going to look like. So I don't have a good answer today, but it's a really important question and one that we have to be thinking about right now.
So the body will create antibodies against AAV74, right? Mm-hmm. So will body accept AAV9?
I think that's a really good question. I think that the honest answer is that I don't think we really know. So AAV74 and AAV9 are different. They are different vectors. But I think one thing that we also have to keep in mind is that you could get exposure to AAV kind of throughout your lifetime. So I think one thing to think about is the span of time in between, kind of the hypothetical case where if someone was treated with, say they were treated with AAV74 and there was a span of time, that person would still, even if it was theoretically possible to be treated with another vector, and it might be, you would still have to be screened to see if there were neutralizing antibodies to the second vector. So I think it's not as easy to answer because of that span of time when there's so many things that can happen in a person's life. It's hard to say if it would be even necessarily directly related to the fact that the person had been dosed with gene therapy the first time, or if they were exposed, in a different way. That would be a really hard question to answer. But again, a really important and pivotal question, and I think we all need to be thinking about what happens to patients later. We can't wait and start thinking about that in 10 years. We have to start thinking about that today. So thank you so much for bringing that up.
So what is the virus you are using for CRISPR-Cas9? AAV74 or 9 or something?
So we actually haven't decided finally, but we are about to decide which type of virus we're using. So I cannot give you the answer, but we are evaluating, and it's probably one of the viruses that has already been mentioned, but we haven't actually made that decision. But redosing, it also depends on the strategy, right? And because obviously for gene editing, we are permanently changing the gene, right? So it's not losing anything. We are permanently changing the gene, right? So that could be different in terms of if there needs to be redosing and the time spent before there needs to be redosing.
Hi. Thank you all for coming and sharing with us. We know the competition and to get out and to perform. My question is regarding the criteria. I see that it's mostly young cohorts, ambulatory, with the exception of originally Solid, who came out and allowed non-ambulatory up to 18. But when they got stopped and restarted, I believe you guys went to just young the young cohort again, and I just wondered if you were going to open it up again to non-ambulatory. Absolutely. That's a very important point. The study was always designed and configured to assess SGT-001 in both ambulatory, younger boys, 4 to 11, and also adolescents who were non-ambulatory. And our commitment to that has not wavered. We have decided to go to younger, smaller patients initially after the experience with the first adolescent patient. But that by no means means that we're not going to return to adolescents when we've accumulated the safety and other data that we need to evaluate that and go back. So that's still in the works, and we're as committed as ever to have that happen. But we're starting where we're starting now to inform how we approach then going to adolescents in the continued part of the trial later, too. So I guess the follow-up would be to you and to the others, is there a plan to open it up to non-ambulatory sooner than later? Like, I don't know, like, are you waiting a year and working with young cohorts, and is everyone else going to follow through and open it up to non-ambulatory?
So I could say Pfizer has plans to look at not only to broaden as much as they can in terms of both younger and older. We're still actually meeting like every day to say, how can we accelerate, how can we accelerate? And so it is in our plan, but how soon, I think, we still need to work out. And in part, I think some of the challenges, as we've mentioned before, is the manufacturing. And so trying to balance how much data we can get as early as possible with, because these are weight-based doses, you know, the heavier or the bigger the patient, the more material is needed. And so, like, how can we see as much as we can in younger boys or in as many patients as possible and get some answers before we expand is also some part of the driver. Where they're not that that's ideal, but that's part of it too. So,
Sarepta also is similarly has plans to very soon, hopefully very soon, to be expanding. Our goal is to kick off a registrational trial before the end of the year. And then shortly after that, what we hope to do is to expand and to be testing our gene therapy in different populations. Our CEO has always said very strongly, we don't intend to have a therapy, or our goal is not to have a therapy that treats patients who are 4 to 7 years old. We would like to treat patients who are older, younger, who are heavier, ambulatory, non-ambulatory. And so that means that we need to be, all of us need to be thinking about how to do that in the space of a clinical trial, because we know that access is also important. And so I think, you know, I just echo what Jenny and Tara have said, that that's something that's on our minds a lot and something that we also hope to be doing soon. Great. It's an important question.
Hi, thank you to all the panelists. I guess for full disclosure, I'm a founder of Solid, and I also have a son with Duchenne who's 8 years old. Something that resonates with me, especially with my son, is the days are long, but the years are short, right? And I think anybody, certainly as a mother, can feel strongly about that. And when you have a child with Duchenne muscular dystrophy, I think that resonates even more. So I guess my question to the panel is, if there is one key, you have an audience of patients and caregivers and people that have come on a Saturday night to come and listen to you guys, and you've done a wonderful job explaining each of your programs. I think what would be helpful is if there was one key takeaway you could provide to this audience, whether it be something that, I don't know, whether it's something that you think that they need to know about gene therapy or perhaps a challenge, I would love to hear from each of you. Thank you.
You guys talked a lot today. I don't know for a second. It's a good question. So I think I'm always humbled. I've worked in small pharma and big pharma. I think Pfizer being such a big organization, I think that's something that sometimes works against us, especially when we're trying to be nimble and trying to really make sure that while we're at least operating within the confines of regulatory requirements and such, that we can really be as flexible as we can to meet the needs of patients, especially Duchenne boys. I think what we're trying to do is make sure that, that it is heard throughout the organization internally too, that what this need is. And I think that's something that we're struggling with constantly. So it's not just about how can we move faster, but making sure that the need is really heard. I think for us, I know we're not really supposed to talk about manufacturing, but I think one thing we can leverage is being so big and is trying to meet the needs of ultimately, if we're able to get to market, to be able to meet the demand. And so the amount of investments that we've made internally, I think, to try to develop manufacturing within house, I think, is something that I think demonstrates Pfizer's commitment to Duchenne and to finding a cure, hopefully, ultimately. Thank you.
I guess, Annie, we're all very much on the same page as far as the tremendous depth of commitment to doing something absolutely transformative for Duchenne. This is not about small measures. This is about shooting for the moon, really doing something based on the technology that's available and maybe the next best one that's coming to really do something about this disease, to really kind of hit it out of the park. We are committed in all of our resources and in all of our hearts to do this. And that's what I want the community really to know, that we are in this with you and we're bringing everything we can to bear on it to be transformative. So.
You were both very eloquent. What keeps coming to my mind are just two words: urgency and responsibility. And I think those are words that are echoed at Sarepta these days, that we have had early clinical results that have been very exciting, and there's a risk of overinterpreting those, but we also don't want to dampen hope. So we are just thinking very, very carefully about how to move forward urgently but responsibly, and how to be thinking about how to best get our therapies to everyone, including people who today have neutralizing antibodies or who today live in a country that currently doesn't have some of the access that other places in the world do. And I think that it's an important thing for us to be thinking about as industry, and that it's an incredible thing to have a community like this who is able to so eloquently and beautifully say other things that we should be thinking about that maybe we haven't heard before. So I guess my takeaway would be that we want to continue listening, and thank you so much for sharing what you've been sharing, and that we will continue to move forward with urgency and responsibility, not just Sarepta, but I'm sure everyone on the panel.
Very eloquent also.
And I'm cognizant that we're getting near the time. We'd like to have Jesper kind of give his thoughts as well before we wrap up.
So I changed my life to help cure, not cure, because I'm allowed not to say this, but help this disease. I came from the most successful biotech company in the world for many, many years. It's in New York, Immunogenicity Pharmaceuticals. I left that company to join Exonics to try to do something really unique and special. And my hope is that we can we are very small, we will continue to stay really small, we will be doing the best science we possibly can, and we'll focus on Duchenne mainly, but other neuromuscular diseases as well. I think we have outstanding scientists. It's all science-driven. There's a million things to keep them awake because it's complicated and many things. You heard some of them here. But what I can promise is that we will do our very best, do the best science we can, and then we can take it as far as we can, and that is really what it's all about, the best science, and we have the best scientists associated with the company that can help us pull this out through. So that's really what it is. There will be new challenges that I don't know about that I will have sleepless nights over, but that's okay too, and we will work through them. So hopefully come up with something unique and special.
So I have, is this on? I'm not sure. One really quick question for you guys. What can we as patient community do to accelerate getting these drugs approved and accessible to our families.
Poor Tara.
Goodness. So I think it's been, we've been so grateful, especially through Catherine Beaverson, our patient advocacy lead, to be able to work with each of you and to have a constant conversation as we're thinking about our designs or our plans, and that's across many advocacy organizations. And so being willing to continue to talk to us, and for when we think we hear you, we come back and we say, did we get it right? And you tell us, no, we didn't. I think that's great, because it's not even just in terms of all the hurdles, it's not even just in terms of regulatory approval, it's also then how are we going to pay for it? How are the payers going to support it? And so what's clinically meaningful, and understanding those types of pieces. And I think there's also some efforts underway, at least within Pfizer, to try to explore some more objective endpoints and maybe work with some advocacy organizations to help really develop those quickly too. I think those are just some of the things, but just allowing us to, even if we clumsily, get something wrong, to kind of keep us in line, I think is great, because it's not just about convincing the regulators, it's also about convincing the payers and making sure that we are providing something that is beneficial to these boys. I don't know.
Yeah, that's good.
Just, I'd like to add just one thing that I've seen about these panelists, and even from the Duchenne Exchange Group, and I think it's unique, is that these guys reach out and then ask questions for your group and your caregivers and your patients that are signed up on that webpage, and I think that's unique. So I think that it's amazing that these groups have all been represented in doing that. So I would actually say that these groups are kind of in the head of the pack as far as soliciting feedback and from patient-reported outcomes from trial data, but also from just talking about them before. What are the obstacles to reduce patient burden and to make it easier for folks to get in? And I think that's a critical thing, because a lot of people don't do that. But I think as a parent of a child that has muscular dystrophy, you're maybe a little more tuned to that, rather than maybe someone in the industry who might not get that. So I just applaud every one of these groups here.
So I have one thing. I'm a scientist, so I always think about science. And even though we think we understand the disease, life taught me we don't understand everything about the disease. So I think at least what you can help me with is to understand some more of the basic biology of the disease. We talk so much about serotypes, are they AAV positive, are they not positive, these kind of things. But what is really facts? What do we really know? And I think it would be so helpful for all of us to actually understand how many Duchenne patients are actually positive for AAV, what type. We actually don't know. I don't know, at least. Do you know? I don't. And so, establish some of these basic parameters that will allow us to better understand how to treat, how to develop new vehicles for delivering our microdystrophin, our scissors, or whatever, these kind of things, to really think out in the future, because I don't understand it, but I'm committed to try to help understand it and try to develop therapies that are suitable for everybody as much as we possibly can.
Challenge accepted.
Good.
We'd love to work with, and just for the audience, and we're past our finish time, but so I've had the pleasure of working with all of the companies involved here, and I can tell you without doubt, Pfizer's a big company, smaller biotech companies, but even within Pfizer, the rare disease group is a very small company and feels like it. But every one of these companies, you really don't know how committed they are to this patient population. And we've celebrated together and we've cried together. And believe me when I say, when there are roadblocks, they suffer almost as much as we do as parents. And so I know it's a business, but I just want you to rest assured that they really do think about this a lot, and they're very, very motivated. And so I think we are done now. And and again, thank you so much. It is Saturday night, and I appreciate all of you being here. I appreciate the work and the dedication that you're doing to help our kids. Thank you so much, Dr. Huml, for coming up and hosting this panel. It's been really, really informative. And again, thank you to Dr. Wong and all the physicians that came. Thank you so much.